through. So, thanks, Aggie, for hitting record. So, welcome everyone to the final BBI seminar of the year. This is we're really fortunate to have Michael Levin here today. He's going to be telling us a little bit about his research. But before I introduce him, I just want to quickly go through some of the administrative type things. So one of the things is that we basically collate questions that you might have for Mike through a Slido website. So if you type into Google Slido, you'll be taken to a website where you can type in this number, which is in the chat. So this is 513306, and you'll be able to then post questions or upvote other questions that you want to be asked at the end of his talk. Mike has a lot of stuff to go through, so I'm going to be very, very quick now. So to introduce Michael Levin, so he's a distinguished professor in biology at Tufts, and also an associate faculty at the Weiss Institute at Harvard. He has lots of awards and prizes, but one of the reasons I'm really excited that he's here is he does some, I think, super creative research around basically trying to understand some of the biophysical mechanisms that kind of cells and biology in general uses to make decisions around the shapes and patterns that it forms. And he's been doing some really, I think, wonderful work around thinking about how cells as collectives think in some ways, and some of the approaches is to those sorts of problems, and actually not just looking at cells, but actually getting them to do things in different ways, so kind of manipulating those systems. So without any further ado, I'm going to hand over to Michael to tell us a little bit more about some of the things he's been working on.
Thank you so much, and thank you for the chance to share some ideas with you all. I'm extremely excited to be here this morning. Can everybody see my slide?
Yep, that's great.
Perfect. Okay, so just two things. If anybody wants to contact me afterwards, these are the two websites that you can reach me at. All the primary papers, the data, the controls, everything is on the site. And I have to start with two disclosures, which are two spin-off companies, Fauna Systems and Morphoceuticals Inc., which are part of this work. So here are the main points that I'd like to transmit today. First of all, that for the biologists, that fundamental advances and new biomedical approaches are going to require not just understanding the molecular mechanisms by which these things work, but actually understanding the collective intelligence of cells and tissues as problem-solving agents in various types of spaces. And I'm going to try to convince you that a key medium for those kind of computations is something we call non-neural bioelectricity. It's basically a kind of software layer of physiology that sits between the genome and the functional anatomy. And that I think that cracking the bioelectric code, the mapping of this software layer is really going to enable a whole category of so-called electroceuticals for novel applications in birth defects, regenerative medicine, and cancer. And actually, towards the end, we're going to get into the material that's more related to robotics and AI, where I think the plasticity of this biological software, especially as we investigate it through the medium of synthetic organisms, is going to really shed some interesting light on bigger questions, the nature of cognition, the evolutionary origins of mind and behavior, and even some practical implications for general AI. So the biology part of the talk, boiled down to two sentences, is basically this: that like the brain, all of the tissues in bodies form electrical networks that make decisions. Now, these are decisions about dynamic anatomy, and they solve problems. And understanding this non-neural collective intelligence has huge impacts for all these other fields. And I show you this five-legged frog just to remind us that I'm going to show you all kinds of weird-looking animals today. These are not Photoshop. These are real living organisms in our lab that are the medium by which we test our theories and our models. So the first thing I'd like to do is to talk about some fundamental knowledge gaps in biology, and then we'll get to the details of bioelectricity and then talk about the future. So I think one way to envision the end game of this whole field, where is this all going, is this idea of the anatomical compiler. Someday, what you ought to be able to do is sit down at a computer and draw the anatomy of the plant or animal that you want made. So you wouldn't design it at the level of proteins or genes or pathways, but you would simply draw anatomically what you actually want, the way that we do with auto parts and various other things in computer-aided drawing. And so if we knew what we were doing, and if the field was mature, what we would have is a piece of software that would take that description and compile it down to a set of stimuli that individual cells would have to receive in order for them to make whatever it is that you're drawing, in this case, a three-headed flatworm. Now, we don't have anything remotely like this, and it's critical because this problem is basically at the root of almost every aspect of biomedicine, with the exception perhaps of infectious disease. So if we knew how to tell cells what they should build, we would automatically have the solution to birth defects, to traumatic injury, cancer, aging. All of these things have this one linchpin in common, which is how do collections of cells know what to make, and how can we influence that? Okay, and so this is what we're interested in. And in terms of thinking about what are the limitations, why are we not even close to having something like this? Let's just look at some simple knowledge gaps. So here is the larva of a salamander, of an axolotl, and it has legs. Here is a tadpole of a frog, and it doesn't have legs. And so we have the genomes of both of these species. So we know what the genomes are. And now I might ask a simple question. If we combine these embryos early on to make a chimeric embryo, and we can do this, we'll make a frogolotl. And I can simply ask, is a frogolotl going to have legs or not? And we have absolutely no idea. There's no way for us right now from the genetic information to predict something very basic like this. And in fact, this chimeric experiment is simply highlighting something that is true even of each individual species, which is that we can't predict their form from the genome either. Right? You can sort of cheat and compare it to other genomes that you do know what the anatomy is, but we really don't have very good control over the system level properties. And so we want to ask, where does this pattern actually come from? So we all start life as this sort of collective of blastomeres, and then this is a cross section through a human torso. Look at the incredible order, the stereotypical arrangement, the position, the size, the orientation. Everything is exactly in the right place. Where did this come from? We're tempted to say DNA, but of course we can read genomes now, and we know that what's in the genome is actually specifications of protein level hardware. The individual proteins that every cell gets to have, there's nothing directly in there that speaks to the size, the shape, the symmetry type of the organism. So we really need to understand this middle layer. How do these cells know what to make and when to stop? As workers in regenerative medicine, if a piece of this is missing, we would like to know what stimuli would get the cells to build it again. And as engineers, we would like to take one step further and ask, yeah, but what's actually possible to build that's beyond the standard kind of default outcome, right? Given the same genome, what else are these cells willing to build? And this causes us to ask some important questions about the capacities and competencies of individual cells. Now, this thing here is a lacrimaria. This is a single cell organism. There's no brain, there's no nervous system, there's no stem cells or cell-to-cell communication. It's all one cell. This animal, if you're into soft robotics and things like this, you can really be envious of this. We don't have anything remotely this competent in our engineering repertoire. This animal handles its anatomical, its physiological, and its behavioral needs all in one cell. Okay, but what's really interesting is that process can be scaled up, and when these cells join together to make bodies, they don't just lose all this amazing capacity. What they do is they scale it to pursue much larger goals. So instead of local single cell goals, they do things like this. They build these incredibly complex three-dimensional morphologies. And so the story, the standard story of this process is something like this. There are gene regulatory networks, some that turn each other on and off. There are some of these proteins are sticky or they exert force. So there's various kinds of physics that go on. And then when these local interactions take place in very large numbers in parallel, there's this project of process of emergence, and out comes something wonderful like this salamander. And all of these steps are true, they do occur, but there's a fundamental difficulty here and a limitation that we'll explore here, which is that if you wanted to make changes at this end, and you think that our main functional targets are down here, we have a really difficult inverse problem, right? For both for us as workers in biology and medicine, but also for the system itself, for evolution and for development. It's entirely unclear what to change down here to make specific changes up here. And this is a limitation of technologies such as genome editing and so on for goals that we might want to achieve with complex structure. Now, what I want to sort of emphasize is the incredible plasticity and robustness of this process. So here is this salamander. This is an axolotl. These animals regenerate throughout their lifetime. Their eyes, their jaws, their limbs, their tails, spinal cord, ovaries, portions of the brain, and heart. And if you amputate here, for example, the limb will grow very rapidly. And the most amazing thing about this process is that it knows when to stop. When does it stop? It stops when a correct salamander arm has been complete, which raises the obvious question of how does the system know what a correct salamander arm looks like so that it can monitor deviations from that state and then do some sort of means ends kind of analysis to get back to where it is. And when the error is reduced, it stops. So this is a vertebrate that does this. Now the champions of this process are planaria. So these are flatworms. You can see one here. These animals can be cut into pieces. The record is something like 275 pieces. And every piece will grow exactly what's needed to make a perfect little worm, no more, no less. In fact, the new tissue grows, the old tissue actually shrinks down so that the whole thing can be in spatial proportion. And they are, in fact, so regenerative that they are immortal. There's no such thing as an old planarian. Every senescing cells drop off and then they're regenerated and the animal just continues. And you can see the really interesting problem solving here. You make a single cut. These cells back here have to make a head. These cells back here have to make a tail. They were direct neighbors when you came along and separated them, and yet they have completely different anatomical fates. So it's not enough to know the local position of where you are. You actually have to communicate with the rest of the tissue to know what else is here, what's missing, what do you need to make, and so on. Now, in particular, this error minimization process is well illustrated by this, which we discovered a few years ago. This is a tadpole of the frogs, Xenopus laevis. Here's the brain, here are the eyes, here are the nostrils and the gut. And these tadpoles need to become frogs. And when they become frogs, what has to happen is their face has to rearrange. So the eyes have to move forward, the jaws have to come out, the nostrils move, everything moves. And it was thought that somehow what the genetics did was turn standard tadpoles into standard frogs by having a hardwired set of movements, right? Every organ moves the right distance in the right direction, and then you get your normal frog. So we made what we call Picasso tadpoles. Basically, everything is mixed up. The eye might be on the back of the head, the mouth is off to the side, the jaws are, everything is moved. And what we found out is that these animals make pretty normal frogs because all of these organs move around in novel, unnatural paths. And they stop when a correct frog face is made. In fact, sometimes they overshoot and they have to come back. And so what the genetics actually gives us is not a set of hardwired movements, but an error minimization kind of process that has a set point and is able to continue to remodel if started off in the wrong region of that morphospace, is able to solve this problem and still get to where it's going. And this theme of solving specific problems despite perturbations is going to be very important in this talk, and it'll come up several more times. So this raises all kinds of interesting questions, not just about the molecular mechanisms of how this works, but actually about the information processing and what's being measured, well, how do they remember their set points and so on. And so where things stand is that biology in general and biomedicine is very good at getting this kind of information, right? About the molecular hardware, what genes interact with which other genes and so on, protein networks, that kind of thing. But we're a very long way from understanding large scale phenomena like this. This is trophic memory observed in deer first some decades back, where if you make an injury to a particular space, a particular location at this branching structure, this whole thing falls off. And next year, a new set of antlers comes up and it will have an ectopic tine at the same location where the damage was last year. So the cells at the scalp somehow remember the three-dimensional location of the damage, store it for months, and then affect the cell division planes of the bone cells as they make these antlers. To do, what kind of molecular pathway model could even begin to explain something like this, right? We don't even have the proper conceptual models to turn something like this into this kind of thing. So these are the questions, these are very computer sciencey questions that I think are very important to try to understand what's going on in biology. And I think it's a deep analogy, not just a shallow one. because computer science has a lot to teach us. One of the most interesting things that happened there was that this is what programming looked like in the 40s and 50s. So in order to get this computer to do something different, she's reprogramming it by physically changing the hardware. Okay. Now, what the computer scientists figured out is that if the hardware is good enough, and I'm going to claim that biological hardware is definitely good enough, this is not your only option. What you can do, in fact, is control the outcome via inputs or stimuli or experiences. And by taking advantage of the native computational power of this thing, you can program it in progressively more powerful ways that don't rely on controlling the micro level, the micro level details, but actually in the ways that you actually can think about a problem and specify the solution in a much more intuitive way. And to computer scientists and to all computer users, it's obvious that when you want to switch from Photoshop to Microsoft Excel on your computer, you don't get out your soldering iron and start rewiring, right? So we all take that for granted. But in biology, note that in molecular medicine, everything is still at this level. So everything is still about molecular pathways, genomic editing, single molecule approaches. We are extremely excited about the hardware, as we should be. It's wonderful, wonderful hardware. But we really need to move forward and add to this the understanding of the software of what's going on. And so in my group, we think very hard about this feedback loop that enables this process to not just to emergently create a complex shape, but actually to be able to restore it against perturbations. Now, those might be injury, they might be teratogens, they might be bacteria, pathogens, in fact, mutations, all kinds of things. And so actually what is added here are feedback loops, both at the level of physics and genetics, that will continuously try to reduce error. This is in effect a homeostatic process. Now, the thing here that's familiar in biology is, of course, feedback loops. Everyone knows about feedback loops. But what's unique are two things. First of all, the set point of this feedback loop is not a scalar like pH or metabolic level or something that we're used to, or blood pressure. It's actually a complex piece of information that's a coarse-grained description of the anatomy. It's some sort of rough description of the anatomical layout so that the system can tell if things are correct or not. And the other thing about this is that in a cybernetic sense, not in a magical sense, it provides the whole process with a kind of goal-directedness. The goal-directedness refers to the fact that there is a particular target morphology. There's a specific state that towards which the system expends energy to try to get to that state if it can. And once it gets to that state, that's when everything stops. Okay, so that's the functional definition of this target morphology. So this kind of weird way of looking at what's normally thought of as simply a feed-forward emergence, example of emergence, has a strong predictive consequence. The prediction is that maybe we don't have to make our changes back here. If we understood the encoding of that target morphology, the way that the set point is encoded, if we find it and decode it and learn to rewrite it, then we could change what it is that the system builds and actually leave it alone, and in the sense of not having to rewire the hardware at all. And so that only works, of course, for systems that have this kind of scheme. And so we've spent quite a bit of time trying to identify those set points and trying to rewrite them. So at this point, I want to show you how that works. And I think this is just one way. And so let's talk about bioelectricity, which is basically neuroscience taken broader and a broader conception of what a neuron might actually be. So all cells in the body receive this kind of information via a morphogenetic field. It's just this three-dimensional field of information, and it comes in many flavors: chemical, extracellular matrix, tension. So bioelectricity is not the only important thing. I have to make that clear because we're going to spend the rest of the hour talking about bioelectricity. But I think it has some special and unique features, which is that it's not just another mechanism. It's not just one more thing that you have to add to biomechanics and gene regulation to understand patterning. It actually gives some privileged access to the computations that are going on in terms of the control of complex structures. And so in the brain, the way this shakes out in the brain is pretty familiar to everybody. So the hardware looks like this. Cells with ion channel proteins that allow them to flip charged ions in and out and thus acquire a voltage state. And that voltage state can be propagated to their neighbors in the network through these little electrical synapses known as gap junctions. So that's the hardware, and this hardware underlies a really fantastic type of software, and that normally, as far as the nervous system is concerned, is about behavior. It's about moving the body through three-dimensional space. And here you can see this isn't our movie. This is a beautiful visualization of the electrical activity in the brain. And the commitment of neuroscience is to this idea that if we understood the decoding, we could infer what the animal was thinking about. We could decode memories, goals, preferences. All these things are actually in the electrical activity that people are reading out. So this is neural decoding. Lots of people are working on this. Well, it turns out that all cells in your body basically do the same. All cells have ion channels. They set voltage gradients. They communicate those with electric synapses through networks. And this system drives a very similar kind of software, although much more slowly, that instead of moving your body in three-dimensional space, what this does is moves the configuration of the body through anatomical morphospace. And that's not an accident. Evolution basically pivoted that ancient system into the control of behavior when muscles and neurons develop. And so we can do something very similar and bring to bear all the concepts and tools of neuroscience and generalize them to try to decode, to do a kind of decoding so that we learn to read and write this electrical information. And so what we developed was a set of tools to do this. So this is a voltage sensitive fluorescent dye in time lapse observing a frog embryo. And it allows us to get basically a map of real-time map of all the electrical states and all the conversations that these cells are having with each other as the embryo figures out which end is which. And then we do a lot of computational modeling, which you can do to figure out why you're getting particular voltage patterns given the channels and pumps that are expressed. And so just to show you a couple of examples of these native patterns, this is a frog embryo putting its face together. It's grayscale instead of pseudocolor. But what you're seeing is, of course, in time lapse, this is one frame out of that movie. And you can see that even before all the genes turn on that pattern the face, here the tissue has already decided where everything is going to go. Here's where the mouth is going to go. Here's the animal's right eye. Here are the placodes. The left eye will come in shortly thereafter. So here's the one eye, and then shortly thereafter, here's the other eye is going to come in. So this is a native, natural pattern that is absolutely required for normal craniofacial development. If you disturb this electrical pattern, the gene expression is altered and the anatomy follows. That's a normal pattern. Here's a pathological pattern. We've injected these animals with human oncogenes. They will eventually form a tumor. The tumor will eventually metastasize like this. Even before any of that is anatomically apparent, you can already see with this voltage dye, you can already see the electrical state changing. These cells are depolarized. It's an early step after oncogene expression. And they electrically disconnect from their neighbors, at which point they basically just revert back to their unicellular past. As far as they're concerned, the rest of the animal is just environment to them at this point. So that boundary between self and external world shifts based on the extent of your electrical network and your communication. When that closes down, that's when you get metastasis and overproliferation. The cells are just, at that point, amoebas doing what amoebas do. So what we've developed are some tools to actually manipulate these electrical conversations. And what we don't do is use any kind of applied electric fields. There are no electrodes. There is certainly no magnetics, no electromagnetic waves. This is purely molecular physiology targeting the normal mechanisms by which electricity is processed. So we basically get to control two things. One is the topology of the network. So these gap junctions, they can be mutated, they can be opened and closed with various drugs. They can be targeted in different ways to control which cells talk to which other cells. And then we can, of course, directly control the ion channels with drugs, with mutation, with light. In terms of optogenetics, we can put down light masks that will impose specific patterns of voltage onto tissue. Okay, so these are the molecular ways we have of perturbing the bioelectric communication. And so now what happens when you actually do this? I'm going to show you a few examples. So here's one example. Here's an early frog embryo. And what we've done is micro-injected into some cells that are going to become gut. So this gut tissue, micro-injected some cells that with RNA that encodes a particular ion channel. And what happens is that ion channel works to set up a local voltage state, very much like what you saw in that electric face that I showed you a minute ago. So that's that kind of eye-specific spot. When you do this, those cells use that bioelectric map as a reference, and sure enough, they will make an eye. And these eyes can have all the same layers that a normal eye will have. So lens, retina, optic nerve, all of these things. And there are two kind of really interesting parts to this phenomenon. One is note the modularity. We did not provide all of the information that's needed to make an eye. Eyes are very complex. We have no idea how to micromanage the creation of an eye. What we did find is a subroutine call. This is a particular bioelectrical state, not of a single cell, of a field of cells, that tells that region you're going to make an eye. And everything that's downstream of that, all the gene expression that has to, and then the cell movements and everything else that has to happen to make an eye is all downstream of that. All we have to provide is a particular trigger. So that's really good news for regenerative medicine. You don't have to micromanage the outcomes. I'm going to show you many examples of that. The other interesting thing is that there are two levels of instruction here. The first is us instructing the cells what they should be. So the blue is a lineage label. It's a beta-galactosidase co-injected with our ion channel RNA that marks the cells that we have specifically modified. So these here, the blue cells here, are the cells that actually got our new ion channel. Now this whole thing is a lens sitting out in the tail fin of a tadpole. And look what these cells have done. The second layer of instructions is that these cells have said to their neighbors, which were not directly affected by us in any way, you're going to work with us and participate in making this nice round lens. So not only did we get the neighbors to do something interesting, but we got them to communicate to their neighbors and take on a function that otherwise, for example, if that didn't occur, maybe we didn't inject enough cells to make a lens, right? But that's okay. These cells recruit their neighbors to get their job done. So that's interesting. And in fact, you can induce many different kinds of organs. You can make fins, although tadpoles aren't supposed to have fins, so that's interesting. You can induce otocysts, extra hearts, extra forebrain, new limbs. Remember, this is all by manipulating the bioelectric pattern. But then there are many organs we don't know how to make yet. So we don't know how to make liver, kidneys, that sort of thing. That's inactive research. Now I want to switch to a different model system to try to understand some more about how the bioelectrics encodes this information. So here's a planarian. If you cut off a head and the tail, this middle fragment 100% of the time knows what to do, and it makes a head here and it makes a tail there. Now, part of that, it turns out, is because it has this nice voltage gradient that tells it where the head goes and where the tail goes. And we know this is instructive because if you manipulate it and you artificially depolarize this end back here and duplicate that pattern, you can get two-headed worms. Perfectly viable, or in fact, you can go the opposite direction and get no-headed worms. Okay. Now, one can ask an interesting question. If you make this kind of two-headed worm, and then you amputate the primary head, and you amputate this ectopic secondary head, and you let the thing regenerate in plain old water, right? So no more manipulations of any kind. The genetics are untouched. We haven't edited the genome. There's no transgenes. The prediction would be, the standard model would predict that, well, surely then you'd go back to being a normal worm because this ectopic reprogrammed tissue is gone. The genetics are wild type. So of course, this middle fragment should just make a normal worm. But it turns out, once we understood the circuit a little better, it turns out that there is there's a stable attractor of this electrical circuit that corresponds to a single head state. And that's those are your normal worms. But in fact, there's also a stable attractor down here at the double head state. And amazingly enough, that's exactly what happens. If you cut these two-headed animals in perpetuity, as far as we can tell forever, they will continue to remake two-headed animals. Now, we can also we can force the voltage back to normal. So what you have here is the basic properties of a memory. It's long-term stable. It's rewritable by brief transient changes to the real-time electric state. And it has discrete outcomes. And I'm going to show you in a minute, it has latency. But think about what this really means. Again, no genetic change. It means that the information about how many heads a planarian is supposed to have is not, strictly speaking, hardwired in the genome. What the genome does is gives you a really interesting piece of hardware that by default will have the dynamics that cause one head, but it's actually reprogrammable. And once you've made the change, it stays. It stores the new pattern and is perfectly happy to reproduce it until you go back and you change it. Okay, so the latency that I'm talking about is seen here. this is an even more interesting, I think, example. Here is a one-headed planarian with the correct gene expression only on one end. And here's what the voltage pattern looks like, and it makes a one-headed worm. Now, this planarian, again, normal anatomy, normal gene expression. This guy makes a two-headed worm. Now, why would that be? Because in the meantime, what we had done is change this voltage pattern to be duplicated like this. Now, you can see it's kind of a messy, the technology is still being worked out. But what's amazing is that this bioelectric pattern is not the map of this animal. This bioelectric pattern is the map of this animal, meaning that a single normal planarian body can store at least one of two target morphology representations of what a correct planarian should look like. Remember how we got here? We were looking for the bioelectric encoding of the state to which the cells are going to work upon damage. And I've shown you that that can be rewritten to one or two, to one head or two heads. And so what this means is that the bioelectricity is certainly not just a readout of what the heads are doing, which you might conclude from this experiment. You might just say that, well, the bioelectricity is just showing you where the head would normally be. No, actually, this is the beginnings of a counterfactual memory. If you're interested in basal cognition, where do our cognitive capacities come from? This is a very simple example of holding on to a description of events that are not true now, but they will be true if you get injured. And if you get injured, this is the pattern you're going to make, okay? And so we are making all kinds of, sort of conceptual efforts and computational models to try to unify the biophysics of what goes on in these electrical circuits and the state space with approaches that people take in artificial neural networks and other connectionist kinds of fields to really try to understand what does it mean for a network to store memory? What does it mean to be able to recover from error, to regain information once you're damaged, and all of that? There's really amazing isomorphisms here with fields in AI and robotics. Now, I've shown you can make multiple heads, but you can actually go further than this. And you can, as it turns out, you can make heads that belong to other species of animals. So here are planarians with a triangular head. For about 48 hours, you cut off the head, and then for a couple of days, you prevent the cells from electrically communicating with each other. Then you withdraw the agent that blocks the gap junctions, and the animals regenerate to sometimes in a stochastic way, sometimes to the normal correct triangular shape. But sometimes they make round heads like this S. mediterranea, or they make flat heads like the P. felina. Now, it's not only the head shape, it's actually the shape of the brain and the distribution of stem cells. That's exactly like these other species. And the distance between this guy and these is about 150 million years. The frequency of getting these other head shapes is proportional to, in fact, the evolutionary distance between these animals. So you can see here that what you can do by blocking the ability of that electric circuit to occupy the correct region of morphospace, you can actually force it into other regions that evolution has already found and is using them, so these other attractors belonging to. So when you cut, you sort of raise the temperature, right? In dynamical systems speak, you raise the temperature of the system, it comes back down, but it doesn't always come back down to the correct attractor. Okay, and you can go beyond this, and you can actually reach regions of morphospace that, as far as we know, evolution doesn't use, probably because they're not ecologically competitive. You can make standard genetically wild-type planaria into these spiky forms, into these cylindrical, so radically change the body plan symmetry type of this animal, or kind of combination forms. Okay. And these, all of these, the point of all of this is twofold. One is to understand where body plans come from and how collections of cells decide and implement very specific outcomes under perturbations. And of course, there's the biomedical angle. So what we would like to do is to communicate with these cellular collectives and force them to, or encourage them to take on outcomes that are biomedically useful. So here, for example, is a frog. Frogs don't regenerate their limbs, unlike salamanders, very well at all. You cut, and 45 days later, there's nothing. We came up with a bioelectrical cocktail that triggers a grow a leg here state. And what you see here is that, all the pro-regenerative genes start to come on, and immediately you start to get toes and a toenail, and eventually a pretty respectable frog leg. And you can see here it's both touch sensitive and motile. So this is just the beginning. of this work. And in fact, in the most recent work, which is going to come out in a couple of weeks, we show that a 24-hour stimulation of this of the wound with a specific cocktail gives you over a year of leg growth. You know, again, it's a trigger. It's not a micromanagement. We don't we don't micromanage the leg over that year. You just leave it alone. Now, one interesting thing here is that, and this is going to come up again in a couple of minutes, is that it actually can reach the same goal by very different means. So when it regenerates this leg, what you notice is that it is not following the normal developmental trajectory. This is not just a repeat walk through the morphospace that it walked when it was developing, because this is what a normal Xenopus leg, how it normally develops. It's a paddle and so on. But this develops much more like a plant. It's basically growing this middle structure, and then off of it start to branch these things. So there's more than one path, of course, through morphospace, and the system will find some other way to get there. Now, and we'll get back to this shortly. Now, how much of this applies to beyond frog and worm? Of course, I've shown you lots of examples of these things in so-called lower animal models. All of this is extremely highly conserved. Evolution discovered all this stuff around the time of bacterial biofilms. There's some beautiful work showing bioelectric signaling in bacteria. And human channelopathies tell us that this is exactly that's been conserved all the way through evolution. So human mutations in various ion channels give birth defects of anatomy. And so we've done work in human MSCs and cardiomyocytes, and you can control the kinds of things that you can expect to do in vitro. But I think the true power of this thing is actually to set large-scale anatomical structure, not individual cell types. So this is not about making specific types of cells out of stem cells. This is about setting large-scale anatomy. And so what we're doing now, and this is in collaboration with David Kaplan's lab at BME and our spin-off company, Morphoceuticals Inc., we're working to take what we've learned in the frog and now into mammals. So the idea is, can we use David's wearable bioreactor to deliver an ion channel drug payload to shift the state of the wound cells towards rebuilding a limb instead of the normal, protective scarring and all of that. And so stay tuned for that. We're still in progress there. So one thing that's important to point out is which a lot of people ask about, is the molecular mechanisms. Now we know, for example, we know how the voltage changes talk to the nucleus. In other words, how does voltage change turn genes on and off? There's about half a dozen known transduction mechanisms. So we know that on a single cell level. And we know tons about the genes that are downstream of this. So there are conserved, through frog, axolotl, and human, there are conserved downstream targets of bioelectrical change, which are all the pathways that you're familiar with. So hedgehog, notch, BMP, Wnt, FGF, all of these things are downstream, right? So we know those molecular details. But this is what's important to point out, is that this is ultimately very unsatisfying, because what it doesn't explain, it explains single cell properties, and that's nice. But what's left is actually the large scale decision making that is important to understand what it's growing, why it's remodeling, and why does it choose to stop. And this is critical for the control problem as well. And so this is really similar to the multi-scale problem in neuroscience, where yes, you can track the individual events at a single synapse or even a single receptor, neurotransmitter receptor. But what you'd really like to understand are the large scale properties, such as decision making, perception, goal-directed behavior, and so on. And so what we're trying to do is to build these sort of full stack models that go all the way from the molecular network that tells you which ion channels are going to be expressed, then through the tissue level model that shows that under specific electrical states, how they form and how they change over time, and then all the way to large scale anatomical patterns of the whole body that will in turn allow us to infer algorithmic types of models, which make it much easier to make rational changes, right? So all of this has to basically we have to walk this whole path, and we have these. There's this amazing simulator, for example, created by our Allen Center member Alexis Pytak, which allows you to, in every single cell, run a model of an electric circuit and ask global questions like, if you were to cut it into pieces, why does it in fact reestablish the correct electric gradient in every little piece, right? So these sorts of scaling models and scaling thereof are essential to understand what's going on. And the other thing that's essential is machine learning, because the discovery of these models and the discovery of the interventions, what do I need to tweak in this particular circuit to get it to do something different, are probably beyond, in the general case, almost certainly beyond the capabilities of humans to just look at the circuit and say, oh yes, if I tweak these three things, then it's going to do the right thing. So what we've done is we've developed some machine learning tools to, first of all, infer models that match behavior and functional behavior like regeneration, like remodeling, and so on. So infer this not from big data sets like genomics or transcriptomics, but from functional data sets that are in the literature, as in I changed this and this was the outcome, right? So to learn from this and to actually try to make a model of what's going on, basically what we scientists are trying to do all the time. And then actually to use those models to infer interventions, right? Which nodes would you tweak and how to get specific events, to identify really potent high-level control nodes, such as we found in the bioelectrics. And so all of the software is all available for free, and there's papers that describe how to use it and so on. And so just as an example of this, here is because it's really important to understand that this field has moved way beyond where we started almost 20 years ago in mapping the shape of possible deformations that you can make. So this is not about making monsters or weird looking frogs. This is about getting rational control over growth and form. So here you have a normal tadpole. Here's the forebrain, midbrain, and hindbrain. And if you expose these embryos in an early stage to alcohol, nicotine, various teratogens, or in fact mutate things like very important genes like Notch, right? This important neurogenesis gene, you get a really powerful brain defect. So the forebrain is gone, the midbrain and hindbrain are basically a bubble. The animal has no behavior, terrible brain defect. So what we were able to do, again, in collaboration, was to with Alexis Pytak and others, was to build a computational model of the bioelectric pre-pattern that determines the correct size and shape of the brain. Look at the way that bioelectric pre-pattern is altered by mutations, by drug exposure, and so on, and then ask the model a simple question: given that your voltage is wrong, which channels could I open and close to get back to the correct state? Now, that's not anything that, in the general case, that we'll be able to do just by inspection. You need computational modeling for this. And the model actually told us there's a really interesting particular ion channel that what it would do is it would serve as almost a contrast enhancer. It would sharpen the borders that are really in these cases that are really sort of wiped out. And once it told us that, we simply went and found a drug, an existing drug, a known drug that opens that channel. And sure enough, it amazingly, it actually does exactly what the model predicts, and it rescues not only brain structure, so here's the normal brain, but brain gene expression and function, their IQ. They get their learning rates back to to levels indistinguishable from the wild type controls. So this is, to me, this is kind of a striking example of using machine learning to infer very actionable interventions that induce outcomes that we would have never been able to micromanage, right? The brain is a very, very complex thing. So this, I think, is kind of the future of this field, is to use, to develop software tools to design electroceutical cocktails so that you start with a disease state, you use the model to ask what channels and pumps can be targeted to fix it, and then you use the massive toolkit of available, already human-approved ion channel drugs to try to reverse it. And so we have already, this is in collaboration with Jack Tazinski's lab, we already have so the beginnings of the software that you can go and play with that will help you take your cell tissues of interest and look to see what your various control knobs are and which drugs actually can be used to target them. And this computational part is really what needs the most effort now. So that's where I think a lot of the merger of biophysics and machine learning is where it's going. So in the last few minutes, I just want to talk a little bit about what I think the future holds beyond regenerative medicine. And here this is the information that I think might be of interest to roboticists and computer science folks. I want to show you a new model organism. So here it is, it's aqueous, it's moving through a volume of water with some particles in it. And so there it is. You can see here they have different kinds of motions. They can go in circles, they can go back and forth and sort of patrol certain lengths of areas. Here's some tracking data looking at them interacting with each other, right? So this one's going on kind of a long journey. These are interacting with each other. So they have all kinds of movement through the water. The way they move is they have cilia. They have these little hairs that propel them through the water. Here's an example of one doing a maze. So it moves, this is a still water maze. There's no water flow here. So it moves forward, it takes the corner without bumping into the opposite wall. At this point, for whatever internal reason, it decides that there's nothing here and goes back where it came from. So again, just one more time. So there are no gradients, by the way. This is just pure water. So it goes forward, and there's some internal dynamics that at some point cause it to turn around and go back. Okay. It also has the capability of self-healing. So here, after it's been cut basically almost in half, right? Look at the force that it generates through that hinge to come and fold itself back up into the original shape. And in fact, what these flashes that you've been seeing are, this is imaging calcium signaling. So this is exactly what you would see if you were to look down with calcium signaling on various types of brains and nervous systems. This is a kind of a readout of the computations that the cell network is doing. The interesting thing here is that there are no neurons here. This is just skin. These are just epidermal cells. And what I've been showing you, in case you're wondering what this is, what animal this is a proto-organism that's never existed on Earth before. This is a xenobot. And the way that xenobots are made, they're named this because Xenopus laevis is the name of the frog, and then it's a kind of biobot. What happens is we take an early frog embryo, we take the animal cap cells, which are the skin, fated to be skin up here, we dissociate them, put them into a little pile, and we basically give them the opportunity to reboot their multicellularity. So we ask a simple question: if I take standard cells with no genomic editing, no transgenes, I take standard cells, liberate them from the normal influence of their neighbors that are telling them to be skin and sit quietly on the outside of the tadpole and keep out the bacteria, and I put them in a new environment, and they get the chance to see what they will do. Now, they could have done many things. They could have formed a flat monolayer. They could have crawled off. They could have died. Many things they could have done. What instead they do is they work together. They coalesce, and I'm going to play this again. They coalesce, and they actually, over the period of about 48 hours, they come together and they actually form this new kind of creature, the xenobot that has spontaneous motion. It has the ability to self-heal and to do various things that I don't have time to go into now. The amazing thing is that these xenobots have a completely normal frog genome. If you didn't know what they were and you came across one, you might be tempted to sequence the genome to see what animal you're dealing with, and all you would see is 100% normal frog. And one of the interesting things about it is that these the xenobot bodies in their behavioral repertoires have no straightforward evolutionary backstory. Normally, if you have an animal and you ask, well, why does it jump, and why is it a certain color, and why does it have different sensory capacities, the answer is always the same. Well, because for millions of years, its ancestors were honed and selected for these particular properties. Xenobots never existed before, and within 48 hours, they're able to take their genomically specified hardware, things like their cilia and various other capacities, and to use them in a completely novel way to be a new kind of a coherent organism in a new environment and solve problems, morphological problems, behavioral problems that is completely new, that they were never directly selected for. And so I think, and their cognitive capacities, we don't know. We're just starting to understand, do they have memory? Do they have preferences? What can they actually do? I mean, their skin, we think they could probably do a lot, but we don't know yet. What I think this is telling us, much like the many other examples I showed you, such as the tadpoles that still make a normal frog even from a scrambled position, evolution maybe isn't just finding solutions to specific environments and specific fitness functions. What it seems to do is create hardware that exploits the laws of physics and the laws of computation to solve problems in new ways. So here, for example, is the developmental sequence. So here's a genome, and it can go probably many different ways, but here are two ways that we know these cells can go. They can make a nice embryo. So here's a nice frog embryo. at the organogenesis neurula kind of stage that makes a tadpole, and then here are the tadpoles running around and doing things. Or some of these same cells can go here and they make a xenobot that looks like this. If you let it grow and you feed it and you sort of let it develop, it acquires this interesting new, weeks later, it acquires this interesting new shape. That doesn't look like anything in the normal frog sequence. And it makes xenobots that have different behaviors. And one of the behaviors it has is it can make more xenobots. So what happens is, this is the latest paper that just came out on kinematic self-replication. What happens is, if you put loose cells of which the xenobots are made in the environment, what the xenobots will do is actually go around and collect them into piles. Okay, so you can see here it forms these little piles, and then as the piles get bigger, it sort of just keeps working to make them bigger and bigger, and they can make multiple generations. And so we've seen them make several generations. Bots make bots, and the new bots that they make will also go around and collect the remaining cells and make new bots again in several generations. So what you can see here is that we take these skin cells, we liberate them from their normal environment, but we also make it impossible. So we don't add anything, but we take away some constraining influences with the rest of the cells. And in particular, we take away their ability to reproduce in the normal way. And keeping on this theme of getting to the same goal through completely different means, they've apparently found a way to at least partially replicate in a very different way that neither they nor, as far as we know, no animal does this, make copies of itself when the normal way of reproduction is not available to them. Okay, so this whole angle, I think, is very new and interesting. How does evolution give rise to machines that solve novel problems that not only they've never seen before, but neither has that evolutionary lineage, right? Where does that novelty come from? So this is there are many examples here in biology, right? We know that if you amputate at different levels, it will grow only what it needs. In this case, this is a kidney tubule in a newt, a cross section. If you make the cells gigantic, they will still make the same lumen. If they're big, they would just use fewer cells. If they're very big, a single cell will wrap around itself and make the exact same large scale structure. So different lower level molecular mechanisms, in this case, cell to cell interactions, in this case, cytoskeletal bending, can be called up in the service of a large scale anatomical need. So incredible robustness and plasticity to changes in the parts, right? We don't have any robotics where you can just go and make the parts all different sizes and everything still works because the parts come together in different ways. And this, by the way, is one good definition of intelligence by William James, which is the ability to reach the same goal by different means. So this is what I mean when I say that these things are solving problems as a collective intelligence, but it's not just about behavior in three-dimensional space. And it's not just about the body structure in anatomical morphospace. Cells and tissues also solve problems in transcriptional spaces, right? And we have some very interesting examples that I haven't had time to talk about. And of course, in physiological spaces, which are, all of these spaces are high dimensional. I'm just boiling it down to two. And in fact, even really deterministic, simple things like gene regulatory networks, in fact, have the ability to learn from their experience, several different kinds of learning, including associative learning, and you can see some of that here. And so, what I think is important is that not only is biology multi-scale, and everybody knows this, right? The swarm is made of individuals, which are made of organs, of tissues, of molecular networks. Not just structurally are we a set of nested dolls, but functionally, every layer is its own local intelligence, building and solving problems in various spaces, whether they be behavioral space or anatomical space or even transcriptional space and so on. Every step, every level of organization contains its own goal-directed ability to solve various problems, and it's the interaction between those agents and within the same level and across level that gives biology this incredible plasticity. And we call this a multi-scale architecture. And we think that this is the fact that our robotics doesn't have this is first why robots don't get cancer, right? So I've given talks called Why Robots Don't Get Cancer. This is why, because our engineering tends to be single level. The robot is smart, the individual parts tend not to be. That's not how biology works. And the other implication of all this is that it enables this kind of problem solving in novel circumstances now. And just two more quick things. What's happening in the future is that knowing that biology is multi-scale in this, it has this multi-scale competency, what we can do in engineering is replace any level of a biological system. We can replace the materials, the individual cells, tissues, with other materials which are somewhere on this plane of evolved or designed, right? So they could be natural, they could be engineered, and has some degree of a computational capacity. It could be a passive material, or it could be a really complex kind of thing. And all of these different things are we're already seeing this kind of hybridization. So there are already humans that drive wheelchairs with their brain activity and are integrated with various pumps and things. And all of this basically suggests that the familiar range of beautiful diversity in the biosphere, what Darwin called endless forms most beautiful, really is just a tiny corner of the option space of what's actually possible. Hybrids and cyborgs and every possible combination of evolved materials, designed materials, and software is some kind of a coherent agent in this enormous space of possible bodies and minds. And so this has massive implications for understanding biology beyond the sort of n=1 example that we're given by the path of life on Earth. And it has implications for ethics and how we relate to organisms that don't look anything like us, and in fact, don't have the same kind of origin story that we do. So I just want to close here and just sort of summarize what I've said. Biology is solving problems in many different spaces, and that's intelligence. And the intelligence, being the competent traversal of these spaces, is much older than brains. And this kind of multi-scale competency architecture is what provides robustness, plasticity, and evolvability. And there's this physiological layer of software that sits between the genetics and the anatomy. It's the computational medium of this collective intelligence. It is how individual cells bind their activities towards a larger purpose. It's through this electrical network, and possibly chemical, of course, and biomechanical. That remains to be studied. And we now have the ability to rewrite these pattern memories in vivo, to reprogram the behaviors of this collective agent in morphospace to make heads and eyes and limbs. And new AI tools are coming online to help us address all of this, and will have massive implications in regenerative medicine, but also as inspirations for novel AI platforms that are not based on brain architectures, and new regenerative medicine roadmaps where we will work on incentivizing new anatomical goal states to rewire not to rewire the actual machinery, but to simply to change the goal states towards which these cells work. So I'll stop there. I want to thank the postdocs and students who have done all the work, our collaborators. Always thank the animals, because they do all the heavy lifting for the discoveries that come forth. The funding support of the people that have supported this work, again, 2 disclosures. And yeah, I thank you for listening. If anybody's interested in the details, there are some papers here that kind of go over the bigger conceptual things. And in case you're wondering what 2-headed planaria are like and what they do in their spare time, this is a video for them. This is some people have called these impossible objects. So here they are. So yeah, thank you. And I'll take any questions.
Brilliant. Thank you so much, Michael. That was absolutely amazing talk. It's mind-blowing, the range of things that you've explored and some of the things you've found and stuff like that. I'm joined now by Professor Steve Mann, who's going to help sort of man a couple questions. We've got time for, I think, for 2 or 3 questions. So Steve, is there any that you've particular favorites that have come up, or do you have any yourself that you'd like to ask Michael?
Well, there are a couple on the on Slido, which I should try and put to Michael. I mean, the first one which came up was really about the Picasso tadpole, and how did you construct that in the first place?
Yeah, the easiest way, there are many ways to do it, but the easiest way, and what we did, was to simply change the electrical pattern in that early electric face that I showed you. So that in fact is what happens. If you can use optogenetics, you can use drugs, you can inject channels, but if you disturb the electrical pattern, the whole face will be disrupted. And so you can put the eye anywhere you want, you can put the jaws anywhere you want. That's how we did it.
Okay, yeah. There's another question here about do you think you can extrapolate these findings to more complex organisms, like plants or, to induce flowers, for example? Or are we still a long way from that?
Well, I mean, I don't know, more complex. I think it's not trivial to argue that plants are in some way more complex than frogs. I mean, we can discuss that. But I think the level of complexity, once you get beyond, early bilaterians, the complexity is all there. And in fact, even the human channelopathies are telling us that, all the way across the tree of life, this is well conserved. So yeah, I think, and we're betting that our regenerative therapies in the limb regeneration space will show that, yeah, we can do this in very complex kinds of contexts. Whether this can be done in plants, probably. There are people who do work on bioelectricity in plants, and so I expect it to be possible. Yeah.
It's great. And then just a question about evolution. Somebody asks, how does evolution fit into this approach? Does it directly interact with higher level software, and how might it affect the, in quotation marks, language defining this?
Yeah, that's a super interesting question. So we've studied two aspects of this. The first is how does evolution shape the electrical networks that have these interesting properties? I think one important way to think about this whole thing is the hardware software distinction in computer science. And you have to be careful with these, and I'm certainly not arguing that that biology is like the computers that we use today. But the hardware software analogy is important, because I think what evolution is doing is it's modifying the hardware. So evolution gives us novel ion channels that have interesting properties. For example, some ion channels are themselves voltage sensitive. They're voltage gated. This means that it's a voltage gated ion conductance, which means it's basically a transistor, right? So once you have that, once evolution discovers that, you can immediately make feedback loops, you can make memory, you can make logic gates, and then you can make anything you want. So I think what evolution does is it shapes the hardware in a particular way that once the power is turned on, so to speak, and the system is alive, it will reliably and robustly achieve a particular set of dynamics. And that's the normal. This is why, frog eggs normally make frogs and fish eggs make fish, is because what evolution has done is to hone the hardware so that it reliably has a default outcome. Now, it turns out that what it's also done is created a system that is much more clever and is rewritable. And how that evolves is a fantastic open question. We're just starting to investigate it. But what we do know is that it's very likely that that kind of architecture and that kind of plasticity is actually really good for evolvability itself. So it makes it much easier to evolve things. I mean, think about the fact that these things are not hardwired, but in fact can pursue anatomical goals despite changes. Think about how much that impacts evolvability. It means that if you have a mutation that does something nice, but it also moves your eye, a little bit off, evolution can now reap all the benefits, the positive benefits of that mutation, because the eye will find its way back to where it's supposed to be, right? So it smooths the fitness landscape, and it makes it much easier to evolve. So I think it's this interplay, of evolution honing the hardware, but that architecture actually makes evolution much more powerful.
And when things go wrong, there's a question here about the role of bioelectrics in cancer, solid tumor growth, for example, and could such processes be electrically drugged?
Most certainly. And so we have a lot of work on the electrical control of cancer. Cancer can be induced and suppressed in vivo by bioelectric changes that do not require any mutations, any carcinogens, any of that. So we have normalized tumors induced by human oncogenes in frogs. So we've shown how that works in frogs. We are now moving it to human cells. So we have projects ongoing in the lab. I think it's absolutely possible. Mm-hmm. Yeah.
And the question here, which I find fascinating as well, is the person asks, is there temporal rewiring, for example, acceleration, happening as well while keeping the spatial morphologies? Can you accelerate limb growth, for example?
Yeah, it's a good question. We don't know. I think, at a clinical level, you can sort of think that worst case scenario, if the limbs take as long as they normally do, someone who loses their limb in an accident in their 20s will have you know, a 7-year-old's arm, you know, within 7 years, and you know, children's arms are extremely usable, right? You can do anything you need, and then eventually, you know, you'll have a normal arm. So I think biomedically, I think it's fine, it's okay, but it would be nice to know if time is under these controls. We don't know. We don't know that yet.
Great. Tom, do you have any questions?
This is a question I had when I was listening to what you were talking about. So you kind of mentioned how conserved a lot of these processes are, and that was something that you highlighted. But I was wondering, obviously, the starting point that you're beginning from, the cells that you're choosing to work with, I'm just wondering how much of an effect they have on what capacity you have to explore different functionalities these things might have, and whether you're doing any work to look at what organisms you might want to target based on something about how they're actually, I don't know, the niche that they live in, and how evolution might have shaped some of the actual hardware and software in different ways.
Yeah, no, that's a very good point. And we have a zoo in the lab. We have all kinds of cells that we work with. And I think that the fact that you can make xenobots out of wild type cells, and that you can make worms with other head types, suggests that there's a huge amount of plasticity in what you can do with specific cells, but there may be advantages to using certain cells for certain reasons. And we are also really interested in chimerism and building composite, building collective intelligences that are made of very different subunits that have never worked together before, right? And so what happens when you have a set of cells that want to make a particular anatomy, and they're mixed up with a bunch of cells that want to make a completely different anatomy, right? What is that collective agent going to try to do? And that has a lot of implications. So we're investigating it.
Yeah, no, it's this almost like an idea that you could have. I'm a computer scientist, so I'm thinking of like evolution could have developed these separate programming languages in a way that have evolved, but we don't see them like that, and whether they actually exist, and what that might, how the implications that might have for the sorts of capabilities.
Absolutely, yeah.
Cool. Steve, I don't know, we've probably got time for one more question, I think, because I realize we're running over time. And then Steve, did you have anything?
Yeah, I'll ask one of my own questions then. Yeah, so Michael, what about the set point? What controls that? I mean, as you emphasize, it, you know, finishing the job is really important. Making sure, you know, when you've got the five digits, and it's the set and proportions and so on, the scalar proportions. What defines the set point, and where is that? Where's the feedback of that coming at that stage?
Yeah, so we can see these set points in the bioelectrical distributions, and the question is where does it come from in the first place? So we see them arising all the way from singles, and at least in the frog, you can track them from the single cell. And I think this goes back to the question about evolution. So what evolution does, you can imagine an electric circuit where what happens is the parts are selected such that when the power is turned on, it has a default behavior. It has a stable pattern that it settles into. And that's what I think evolution has done, is it's given a bunch of ion channels such that there's self-organization, there's symmetry breaking and self-organization, like Turing patterns. You can sort of think about it, that if under normal conditions, the electric circuit will settle in a particular pattern with, you know, depolarized over here, hyperpolarized over here, and now you get a head-tail axis, that kind of thing. And I think that's part of what evolution does, is it helps pick parts that under normal circumstances reliably settle into the desired initial part. So there's a lot of symmetry breaking and self-organizing principles here.
But is that because you reach a point of sort of a steady state? Is it energy that really is defining it? The energy distribution across the organism? It's in non-equilibrium states, of course.
Yeah, some of the patterns. I mean, you can think about it this way. Imagine two cells. And there's they express a voltage gated ion channel, right? So let's say one of the cells is a little bit depolarized. Well, that opens the channel, so that makes it more depolarized, which opens the channel, and so that drives this positive feedback loop, and it gets really depolarized, and eventually it'll just settle to the lowest, the lowest depolarization that it can, and it will sit there until something new comes along. Maybe this cell signals it in a way that causes it to close the channel again, and now the thing gets reset. But if that doesn't happen, it reaches a stable point, right? So all of these bioelectric circuits have stable attractor set states.
Mm-hmm, mm-hmm. Great, thank you.
Brilliant. So I think in the interest of time, I'd love to just sit and chat about questions around this for all afternoon, but unfortunately we can't do that. But I just want to thank you again, Michael, for a really fascinating talk. It was brilliant to learn about some of your recent discoveries, and thank everyone for attending, and thanks Steve also for helping to field the questions. So with that, I'll bring this session to a close.
Thank you very much, everybody. I'll see you soon.
Thanks.
Thanks. Bye.
Oh. Awesome. Thank you.